course, it is represented by plants, animals, including the arthropods, that is insects and related organisms. Then among the microorganisms, you have algae, fungi, and protozoa. And the other two branches, the domains, they are also domains are also called as super kingdoms. Super kingdoms are domains, and they are represent. The two branches, two domains represents prokaryotic cellular organizations, the bacteria and archaea, and of course together in the Burgess manual, they are all considered in bacteria only, but in the universal phylogeny, the archaea are made separate due to some structural differences with respect to their cell walls, cell membranes, and other aspects. The bacteria, the first group among them, the bacteria, by definition, they are the unicellular prokaryotic organisms which reproduce asexually by self-fission, that is, which is simpler than the cell division, and which lack sexual reproduction. That's the simple definition we can give. And some general aspects about bacteria in the previous things we have discussed. Bacteria are more numerous and they are metabolically more diverse. They include both heterotrophs and autotrophs, in that both photoautotrophs and chemoautotrophs. All kinds of organisms are found in bacteria. They include both aerobes and anaerobes, including anaerobic respiratory organisms and fermentative organisms. They include the mesophiles, thermophiles, and psychrophiles, and like that. If you consider anything, that is all aspects. For example, with respect to osmotic concentration, so we use the thing, osmophiles. They are also represented by bacteria, which are represented by saccharophiles with respect to sugars, or halophiles with respect to higher salt concentration or lower salt concentration. All that, various, plus so many other things, in the previous chapter we have discussed. So bacteria, so they are unicellular prokaryotic organisms, are unicellular organisms with prokaryotic cellular structure. So with asexual reproduction, they reproduce asexually by simple fission, and they lack sexual reproduction. However, they exhibit three kinds of genetic recombinations or transformations. They are transformation and transduction and conjugation, all these things. And of course, while discussing about bacteria, we discussed the thing, common types of bacteria, in that, like the endospore forming bacteria. With examples, we discussed what are endospores, what are endospore forming bacteria, and all that, So some common features about these organisms and examples also we have discussed. The next we discuss about the photosynthetic bacteria, which is represented by two groups of bacteria. An oxygenic photosynthetic bacteria represented by green sulfur bacteria and purple sulfur bacteria, that is chlorobium and chromacium, which have cyclic photophosphorylation. They use hydrogen sulfide as source of reducing power. So all that, they are anoxygenic. They don't release oxygen. Instead, the sulfur, which is part of H2S, after dissociation of the molecule, splitting the molecule and taking out H plus and electrons, protons and electrons, sulfur is accumulated either inside or outside the cell. That's why they are called as photosynthetic sulfur bacteria. So Few more examples are there for all of them. After every class, you have to go back. Now only option is Google before you, and many e-notes are also available. Soft copies are all online, and specific groups. You get information on the Googles. Note down the examples also. And next is oxygenic photosynthetic bacteria represented by cyanobacteria. So which have which are represented by organisms like Anabina, Nostoc, Oscillatoria, so Calothrix, Cytonema, Tolipothrix, and so many other examples. You will find them if you give search for cyanobacteria. And they have oxygen evolving photosynthesis similar to higher plants. Because why? It is mainly because instead of H2S, they make use of H2O as source of reductants and electrons. So they think, in the anoxygenic photosynthetic bacteria, the resulted sulfur is accumulated, whereas in these oxygenic photosynthetic bacteria, that is represented by cyanobacteria, they resulted oxygen. It is evolved back to atmosphere. During evolution also same thing happened. After the after these cyanobacteria came into existence, then the atmospheric, it became, it was oxygenated. So earlier it was anaerobic, anoxygenic. So after the presence, the entry of the cyanobacteria, slowly the environment outer becomes atmosphere became oxygenic, aerobic, and also the ozone layer was also created after the incoming of these organisms. And many cyanobacteria, of course, they include both unicellular and some of them multicellular also. But multicellular, they lack tissue organization. They don't, because each cells are independent metabolically. And here, so majority of the cyanobacteria, many of these cyanobacteria, they have the ability to fix atmospheric nitrogen in addition to their photosynthetic activity, ability to fix atmospheric carbon. They have both carbon fixing and the nitrogen fixing abilities. So both they have. Some examples, for example, nitrogen fixing cyanobacteria, say Anabina, Nostoc you can give, or Calothrix, Tolipothrix, Cytonema, Apellosyphon, Fissurella, like that. You can go on giving examples. These, there is a particular group where this Anabina, Nostoc are there. They are heterocystis filamentous cyanobacteria. They have a structure called heterocyst, which is a very special cell, a modified vegetative cell, which acts as site of nitrogen fixation. So all of you should go back after this class. Kindly refer for heterocyst in Google. Heterocyst of cyanobacteria. So the structure of heterocyst and their functions, so all that. And these cyanobacteria, they have both photosystem 1 and photosystem 2, the cyclic and non-cyclic, where photosystem 2 is mainly operational, more operative, so which actually the cause for the oxygen evolution, oxygenic photosynthesis. And next you have chemosynthetic bacteria represented by nitrifying bacteria, sulfur oxidizing bacteria, then iron oxidizing bacteria, and similar kind of other bacteria. Where these bacteria, they derive energy by oxidizing reduced inorganic molecules like ammonium or hydrogen sulfide or elemental sulfur or the metals, like iron. So they, by oxidizing, so they derive energy. Examples you have in the nitrifying bacteria, Nitrosomonas and Nitrobacter. Sulfur oxidizing bacteria, you have Thiobacillus. like and related organisms, and iron oxidizing bacteria, ferrobacillate, and related organisms, all these things. So the so the chemosynthetic. Similarly, you also have the stalk bacteria. So they are represented by the thing, like colobacter, uh, hypomicrobium, and so on, where these bacteria, they are attached to the thing, like rock surfaces. Especially these organisms are found in the streams. So they are iron oxidizing. Their stalks are made up of ferric hydroxide. Ferric hydroxide. So the colobacterium, hypomicrobium, and so on, gallionella, and so on. So next you have some fruiting bacteria. There are some examples. Kindly go back and make the notes. Next we have the archaea, a group represent archaea. Archaea by nature, of course, they are prokaryotic organization. They have prokaryotic cellular structure, but they differ in some aspects, especially cell wall composition. Instead of murin, they have pseudomurin. Then, the other aspects with respect to cell membrane structure and so on. And so like by nature, these archaebacteria, they are extremophiles. They are found in extreme environments like highly acidic conditions, highly alkaline conditions. Then at high temperature, hyperthermophiles are also found. So many examples. Then highly anaerobic organisms like methane producing bacteria. So many examples you can give for this, particular things. So methanogenic, then thermophilic bacteria, then acidophilic bacteria, and so on. So kindly at least make five examples for this category: archaebacteria, archaea, some general characters and other things. Next you have the other group, the actinomycetes or actinobacteria. Earlier we were referred to, even now also we use that more oftenly we use the term actinomycetes. So, but in the Burgess manual they are referred as actinobacteria. Actinobacteria, and these actinobacteria, they are a group of bacteria with special characters. Like they are also referred to as sometimes ray fungi. They have they are known to produce very thin tubular structures. Tubular structures resemble the hyphae of the fungi. So they are, but their thickness is 0.5 micrometers, whereas the hyphae of the fungi, it is quite larger, broader. Then of course, except streptomyces, in other especially in nocardia and arthrobacter and related organisms, though they exhibit this filamentous structures, just by little disturbance they form bacillary structures. And actinomycetes, of course, they are known to be involved in organic matter decomposition, humus formation, even humus formation also. And these organisms, of course, by producing antimicrobial substances in the form of antibiotics and other metabolites, they are also responsible for regulation of the soil microbial population. Of course, like they are the source of the antibiotics, industrial production of antibiotics from many of them. It started with Penicillium notatum by Alexander Fleming, penicillin. So now you find hundreds of them from this particular group. So then, of course, these actinomycetes are mainly represented by Streptomyces species. They are responsible for that musty odor when the soil is plowed early in the morning in hot summers. You get that characteristic musty odor, soil odor, that is due to the production of geosmin. an aromatic compound. So the thing, aromatic compound produced by these organisms. Of course, actinomycetes, they are also represented by some pathogenic ones. They are responsible for powdery mildew and other diseases, scabs, powdery mildews, and so on. Next, you have nitrogen fixing bacteria, another group you can give as a unique group of bacteria. So you have already covered endospore forming bacteria, then photosynthetic bacteria, in turn anoxygenic and oxygenic photosynthetic bacteria, chemosynthetic bacteria. Then you had stalk bacteria, fruiting bacteria, orchobacteria, actinobacteria, and so on. Actinobacteria or actinomycetes. Then, of course, together represent by a unique character, that is ability to reduce gaseous nitrogen to ammonical nitrogen. That is, the organisms which can use gaseous nitrogen as source of nitrogen. So with the help of the enzyme, the nitrogenase, they reduce N triple bond N. That's you also called as uh, a diazote, that is N triple bond N. In 3 consecutive steps, it is reduced to 2NH3, 2 molecules of ammonia, with the help of the enzyme nitrogenase. And this nitrogen fixing property, it is only found in these bacteria, not in eukaryotes. It is the unique features of bacteria. And of course, in this you have different kinds of bacteria: free living nitrogen fixers, associative nitrogen fixers, symbiotic nitrogen fixers, and so on. Some of the examples, like you have Azotobacter crucoccum, Azoospermum lipoferum or Brazilians. Then you have Rhizobium, then Rhizobium fascioli or Leguminosarum, and so on. Then Bradyrhizobium japonicum. Then you have, of course, in non-legumes, an actinobacterial genus Frankia is known to form the nodules, like in Casuarina or Alnus, and so on. So they you have the Frankia alni. So then, like that, Azolla symbiosis in the Azolla, which is an aquatic fern in the leaf lobes of this organism, a cyanobacterium, Anabina azole, it lives and it fixes nitrogen. Like that, you have many examples. Of course, these organisms, the organisms which have the ability to reduce gaseous nitrogen to ammonical nitrogen with the help of the enzyme nitrogenase, which is also, this process is also referred to as biological nitrogen fixation. And these organisms, they enrich the soils wherever they grow. They enrich that environment with chemical nitrogen, in addition to their primary function, that is carbon fixation, addition of the organic matter. So these are all some of the properties of bacteria. Plus you have, of course, many other things. Kindly refer and make down the notes. So then coming to some general roles of bacteria. Roles of bacteria to list some of them. These bacteria, of course, since they are metabolically diverse, and so they are involved in the biogeochemical cycles, recycling of the nutrients, various nutrients in the soil, recycling of nutrients in the soil. They are responsible for biological nitrogen fixation. Then, of course, they do add organic carbon to the soil in the form of, in the process of photosynthesis, and many of them are known to dissolve phosphorus, bring phosphorus from insoluble form to soluble form. You call this as phosphorus solubilization. So phosphate solubilizing bacteria, like you have Bacillus megatherium and related organisms. Then you have, of course, antibiotic producing, they are known to produce antibiotics. They are known to photosynthesize. They are known to fix nitrogen biologically. They are known to be involved in biogeochemical cycle, recycling of various nutrients, and so on. Of course, many things you can recollect. In addition, of course, many of these bacteria are also known to be pathogenic, like you have mildews, powdery mildews. So the actinobacterial genera, sorry, the actinobacteria, uh, you know, actinomyces scabies, sorry, scabies. Then you have, of course, Pseudomonas solanacearum causes wilt. Then Xanthomonas campestris, again wilt and blight diseases. Like that, you can recollect many of them, in this particular groups, general roles. The second group, it is fungi. Second group of the organisms in the cell or microorganisms, it is fungi. And fungi, by definition, they are a group of eukaryotic achlorophyllous organisms which reproduce both sexually and asexually. They reproduce both sexually and asexually, and they include both unicellular and multicellular forms. Unicellular ones are referred to as yeast, multicellular ones are referred to as molds. So molds, so the thing, that's the definition. That is, they are a group of eukaryotic achlorophyllous organisms, including both unicellular and multicellular forms, which reproduce both sexually and asexually. Okay, and which are involved in, or especially responsible for, organic matter degradation. And in these fungi, of course, taxonomically, of course, in the like course of the things, development of this subject, mycology, several it got changed, evolution. But however, you can come across, you can so enlist some of the common groups of bacteria. So for example, you have some 4 or 5 groups of bacteria, fungi you can enlist. Mastigophora, the group. Masticomycotina, the group. Then Zygomycotina, second group. Then Ascomycotina, the third group. Fourth group, Basidiomycotina, and the last group is Deuteromycotina, or imperfect fungi. The first group, Masticomycotina, which includes Oomycetes and Zygomycetes fungi. These fungi are commonly, characterized by having aseptate mycelium. Aseptate mycelium, this is the first point. This, Oomycetes and Zygomycetes fungi, which are part of Masticomycotina things, like Oomycetes fungi and Zygomycetes fungi. Like Masticomycotina and Zygomycotina things. Other thing is, they are, majority of them, they are found in moist regions, either in water and marshy areas. And they need, wherever there is dampness, these organisms are more prevalent. They are aquatic, represented by those which are found in aquatic ecosystem, and marshy areas, and in high humidity, high humid forest, deep forest. These fungi are found. Lower fungi, they are characterized by having aseptate mycelium. And in that, the Oomycetes first group, Oomycetes fungi represented by Pythium, Phytophthora, Plasmopara, and other things. They are most of them. So they are the phytopathogenic fungi, which are responsible for this. The diseases like damping off, rotting, and so on. Of course, Coleroga, a particular disease caused by Phytophthora infestans in areca nut. And of course, in this group, you also have some, fungi which infect aquatic algae and aquatic fungi. Kindly, go back and find those examples in this. Next, the second group is the Zygomycotina. They are sometimes referred to as sugar fungi, as they are involved in the, hydrolysis of carbohydrates to sugars. Examples, you have Mucor, Rhizopus, and so on, Zygomyces, and so on. In this particular group, you have two important things. One is the VA mycorrhizae, vesicular orbuscular mycorrhizae, which are responsible for the, uptake of phosphorus, the fungus root symbiosis, and this group of, like mycorrhizal fungi, they are, from this zygomycetes fungi. They are WAM fungi or AM fungi, vesicular orbuscular mycorrhizae or orbuscular mycorrhizae. Examples: glomus, then you have Gigaspora, Acalospora, Entrophospora, Sclerocystis, and so on, Scutelospora, and so on. These main thing is mycorrhizal symbiosis. It benefits the plants, by through their hyphal scavenging system outside the roads which they infect. They form scavenging system. They help in the uptake of water with other nutrients. More prominently, the less mobile nutrients like phosphorus, zinc, and so on. So they are very beneficial. Then another thing is this zygomycetes fungi. It also is, includes many insect trapping fungi, even nematode trapping fungi. It includes, so again, endless the examples for this particular group. Next is Ascomycetes ascomycotina. That is the fungi, the fungi belonging to Ascomycotina and Basidiomycotina. Both of them are referred to as higher fungi, in contrast to Oomycetes and Zygomycetes fungi, which are called as lower fungi, because they usually colonize where there is more moisture, or in aquatic ecosystem, their habitats, as where there is high humid conditions. And another thing is why they are referred to as lower fungi. They lack septa. As a result, a condition called sinusoidic condition is seen in these fungi, that is multinucleate condition. And the next group, Ascomycotina and Basidiomycotina, are commonly referred to as higher fungi. So why they are called as higher fungi? Because the fungi representing these two groups, Ascomycotina and Basidiomycotina, they have a well-developed mycelial, septate mycelial system, and they colonize terrestrial ecosystem, that is aerobic conditions, the aerobic soils, not the aquatic ecosystems. They are found in the terrestrial ecosystems. They have well developed septate mycelium. They reproduce both sexually and asexually. So that's the thing. In that, the first group is Ascomycotina, and Ascomycotina, the name comes from the very characteristic feature of these organisms. They reproduce sexually through the production of the sexual spores called as ascospores inside a sac-like structure called ascus. ASCUS, ascus. You have learned this in your biology thing, pre-university biology also. You must have learned. And Ascomycetes fungi, they of course they are represented by the the organisms, which are, of course, many of them, they do produce antibiotics, like penicillium. They do produce antibiotics. Many of them, they are of course they are responsible for some diseases like powdery mildews and so on, and rotting and other things. Many of them do produce antibiotics. Many Ascomycetes fungi, they are involved in the salt transformations, organic matter degradation and degradation mainly. And this Ascomycetes fungi is represented by the unicellular yeast. Yeast, you are aware, they are known, very important fermentative organisms. They are known to be involved in processes like alcohol production, organic acid production, and so on. Industrially, they are very, very important. Yes, and they are a rich source of vitamins, B complex vitamins. And of course, yes, their function. Many, fungi representing this group, yeast group, of course, are responsible for powder mildew disease also. Um, so next, the next group is Basidiomycetes fungi. So Basidiomycetes fungi, they are those, second group of higher fungi, which reproduce through the production of the sexual spores called basidiospores. These basidiospores are produced on a stalk-like structure called basidium, on a club-shaped structure called basidium. So the basidiospores are produced externally, whereas ascospores are produced endogenously inside sac like structure called ascus, whereas these basidiospores, are produced externally on a club shaped structure called ascus. And of course, you can recall the thing, gills, in mushroom fungi, which are known to exhibit this kind of things, basidiospores, the mushroom fungi represented by agaricus, pleurotus, calocybe, and so on. Boletus, razula, tricoloma, like that. You have many fungi, and these fungi, they produce ascospores. They produce sexual spores called basidiospores, and on gills, on lamellae, so the thin, laminate structures. On that, on a club-shaped structure basidium, they produce these basidiospores. And by nature, these basidiomycetes fungi are powerful lignocellulolytic organisms. So lignocellulosics, sometime I must have told you about this particular cave. What are lignocellulosics? They are the thing, organic residues originated from plants. Since plants are the rich sources of cellulose and lignin, so lignin is found only in plants. Cellulose is also again the plant. So the the plant residues are usually referred to as lignocellulosics. Then the organisms which cause the hydrolysis or degradation of this lignin, cellulose, and related things, so they are called as lignocellulolytic organisms. These mushroom fungi are responsible for the degradation of organic matter. So they are the climax community. So that is seen, during the initial stage of the organic, in the organic matter degradation, responsible for the degradation of tough molecules like lignin, cellulose, hemicellulose, chitin, and so on. And of course, this Basidiomycetes fungi, of course, as you are aware, they produce mushrooms. They act as food for other organisms. Then, of course, they are responsible for the organic matter degradation. And many these Basidiomycetes fungi are also pathogenic, like you have rust, smuts, and so on. So the rust and smut diseases, they are caused by Basidiomycetes fungi. Like that, you can enlist many features, both positive, including both positive and negative roles of these organisms. And the last group in the fungi, you have the imperfect fungi, or the deuteromycotina. Deutero, D-U-T-E-R-O, M-Y-C-O-T-I-N-A, mycotina, deuteromycotina, or Deuteromycetes fungi, the imperfect fungi. And in the course of the, during the development of this, during the initial days of the development of mycology, as a separate branch of biology, at that time, people who are working with fungi, they noticed that some of these fungi lack sexual reproduction. They don't exhibit sexual reproduction in their life cycle as a routine, regular feature. So they call them the imperfect fungi because they lack sexuality. They call them imperfect fungi, and they use the word. However, after a decade or so, other scientists they reported that they do have the sexual reproduction. Only difference is, and though they'll have sexual reproduction, but it doesn't occur in a regular cycle in their life cycle. It may, it is a rare feature. That's why, of course, I know they call, of course, initial days they created a separate thing, for them, imperfect fungi or deuteromycotina. So all those which lack sexual reproduction, mainly coming from ascomycetes and basidiomycetes fungi, they're all brought under this particular group. If you look through, go through this literature, so the mycology, majority of these, genera, majority of the soil fungi, they're all represented by deuteromycetes fungi, deuteromycota, imperfect fungi. Like you have Aspergillus, Penicillium, Fusarium, Alternaria, Helminthosporium, Botrytis. Then you have Cladosporium. Then you have Helminthosporium. Like that, you can go on listing even Rhizoctonia, Sclerotium, and so on. Many of them, you can go Curvularia, Cercospora, and so on. So, all these things, which are there in the soil, and of course, the common soil fungi, which I have told you this list of names, they're all, in this group, imperfect fungi. But because they think, not, the like after the thing, after reporting by certain scientists that they do have the sexual reproduction, but it is a rare feature. Then the fungi coming, enlisted in this group, they gave two names. One name for perfect name, the other name for imperfect name. For example, Fusarium. Fusarium is the imperfect stage. Gibberella, it is the perfect stage. Like that, kindly, of course, at least 5-10 examples. These for imperfect fungi, you can have both the names. Perfect, a perfect name and imperfect. Name for perfect stage and name for imperfect stage. You can enlist, and these are all some of the things. For example, Masticomycotina, which includes oomycetes and zygomycotina, oomycetes fungi, trichomycetes, all that. Then zygomycotina, then ascomycotina, then basidiomycotina, then deuteromycotina, imperfect fungi. This imperfect fungi, of course, they lack sexual reproduction. However, they reproduce through the production of various asexual structures like conidia, mainly conidia forming organisms, conidiospores, through the production of conidiospores, then chlamydospores, arthrospores, oidea, and so on. Various types of asexual spores they produce and reproduce. And of course, after covering all these things, some general information on these fungi, ultimately to list some of the roles of these, some few roles of these fungi. Of course, you can say fungi as a general group. One is they have, through their lignocellulitic activity, they are involved in the organic matter decomposition. That's the first point. Second point, these fungi or mushroom fungi, they are the source of food for other soil organisms. Third, these fungi, they are as involved in mycorrhizal symbiosis, are known to be responsible for the uptake of nutrients, mainly less, mobile nutrients. Then fungi, they are also, they are source of enzymes, like many enzymes, cellulases and different kinds of enzymes, complex enzymes used in industries. Of course, many of these fungi, they are the source of these enzymes. Many of these fungi, they are also known to produce antibiotics, like in penicillium, which produces penicillin. You have many other things and examples. So that's the things. Next, they are the causal, agents of many plant diseases like rots, wilts, then damping offs, rust, smuts, and so on. Varieties of diseases. They may be responsible for varieties of diseases. Of course, they are also as a unique group of organisms. They are also utilized in biological control, like Trichoderma horseanum or Trichoderma viridae. So these fungi, they are actually um, used in biological control. Many insect trapping fungi, they are also utilized in biological control. Biological control of nematodes. Nematodes mainly, they are utilized, and they do cause the thing, like diseases of some aquatic, animals and insects. Their larvae, they are So this is the thing, some thing, like a few information about fungi. And the next group of organisms, it is algae. Next group of organisms, it is algae. So algae, by definition, similar to fungi, you can define, in your own words, fungi are a group of eukaryotic photosynthetic organisms which include both unicellular and multicellular forms and reproduce both sexually and asexually, and are known to colonize both terrestrial and aquatic ecosystems. So this is the simplest definition you can give. And algae, of course, earlier, in 60s and 70s, till the end of 70s, the cyanobacteria, they were also included along with this, like higher algae. But later in the 80s, they were separately designated as cyanobacteria. And these algae, they are actually classified based on their pigment composition. That's very, very important. Here, for the classification of these algae, pigment composition of these organisms, of course, the primary pigment is photosynthetic pigment, chlorophyll, either chlorophyll A, B, or C, and so on. And in addition to primary pigment, they have the secondary pigments, like you have xanthophylls, carotenes, phycobilins, phycocyanin, phycoerythrin, and so on. So the biliproteins, they are secondary pigments. The presence of these secondary pigments, it is taken, it is considered for classifying them in addition to the primary photosynthetic pigment chlorophyll. For example, the green algae. The secondary pigment, here the primary photosynthetic pigments chlorophyll A and B, they are dominant as compared to the other secondary secondary pigments like carotenes or xanthophylls. Carotenes, you should know, these secondary pigments, they are responsible for different colors, especially xanthophylls, yellow color, carotenes, brown color, phycocyanin, blue color, erythrin, red color, and so on. So depending, suppose if Any of these pigments are dominant, that name comes to that group of organisms. In green algae, the photosynthetic pigment chlorophyll is dominant, masking. Of course, they do have xanthophylls, carotenes, and carotenes, but you know the thing is it is dominated by chlorophyll pigments. That's why they are called as green algae. Examples you have chlorella, chlamydomonas, then chlorococcus, like even spirogyra and so on, Cenedesmus and so on, various things are there, green algae examples. And green algae, they are usually found in terrestrial, they are also, they are found in terrestrial ecosystem or in freshwater streams, aquatic ecosystems also. They are known to form, green scums on the surfaces of the pond waters, which are actually, stagnant water bodies. Of course, also found in streams. And many of these green algae, they are also found in the marine water ecosystems also. That is, you have estuarine water ecosystems and in the marine ecosystems. Then you have the yellow-green algae, yellow greens represented by heterothryx and Botrydopsis, yellow greens. So here yellow, I've told you already, the yellow color, it is the xanthophylls. Xanthophylls, they are golden color or yellow colored pigments. Their pigment, they are equally present in addition to chlorophyll pigments. That's why the two names yellow greens. Xanthophylls and the chlorophyll pigments are, so they are in equal, concentrations. And these yellow-green algae are mainly found in terrestrial ecosystems. Next, you have the golden browns, golden or yellow, golden browns. They are also called as bacillariophytes. The diatoms. This group, the organisms representing this group, they are known to have the halved cells. They are unicellular in nature. They have golden colored xanthophylls, brown colored carotenes, in addition to the chlorophyll pigments. And these diatoms are known to have chlorophyll C. Unlike the green algae, which have chlorophyll A and B, and yellow greens also the chlorophyll A or B, but diatoms are known to have the chlorophyll C in addition to the other two pigments, the golden colored xanthophylls and brown colored carotenes. That's why the name golden brown algae. Or they are also the bacillariophytes, or commonly referred to as diatoms, where their cell walls are halved. And then, in one of the previous chapters while discussing, I told you diatoms, the cell walls of these organisms, may contain either cellulose or chitin, or they may be impregnated with some inorganic things, materials, where you call them as frustules, where you use the word frustule. Example, diatoms, their cell walls are impregnated with silicaceous materials. And these diatoms, they are the major primary producers. Like the higher plants in the terrestrial ecosystems, in the deep oceans, the diatoms are the major producers. So the frustules, they are halved, inner halve and outer halves, and they are silicified. After their death, their dead matters are settled down to the seabed. They form what is known as silication, what is the diatomaceous earth you call, which is the rich source of silica. It is mined and taken out and used for various purposes like abrasives, toothpaste making, for, the making of the filters and so on. So they are used. Diatoms examples, you have Acnanthus, Cymbala, and so on. Of course, you should remember in deep oceans, it is the diatoms which are the, so the, which are the primary producers. So near seashore, you have the thing, the seashores, the higher plants. Then you have mangroves, the plants with propped roots, propped roots. Then down, if you go, you will come across some kelps and so on, and the filamentous fungi, the sargassum and such kind of things, which are things. Further, if you go into the sea, they are represented, slowly replaced by, the filamentous ones, which don't form macroscopic structures. Further, if you go miles together in the deep oceans, you come across unicellular algae, mainly represented by the diatoms, yellow greens. They are the source of food for other aquatic life in the deep oceans, marine life in the deep oceans. Okay. Next, you have the euglenoids. Euglenoids are there. They are, of course, the yellow greens are sometimes they represent because euglena through their food vacuoles, they can also digest. Euglenoids, of course, they can be similar to yellow greens. Then you have the red algae. Red algae. In these algae, in addition to chlorophyll pigment, they have the major pigment phycoerythrin, a red colored pigment. That's why the name red algae. Red algae, porphyridium. Then you have, of course, red algae. Then you have the brown algae also. Brown algae, for example, sargassum and other forms. This red and brown algae, they are the source of many complex, the polysaccharides, like agar agar. It is made from these seaweeds. It is made from seaweeds. Then you have, these are all the things, of course, dinoflagellates are also there. And algae as such, the common roles to enlist in them, by photosynthetic activity, they add organic matter. They add organic matter to the soil. So they are the, of course, they have a very important role in the soil forming process, especially deserts to make them, fertile and, deserted environments to reclamation. These algae are used. They are the main source, by their photosynthetic activity. They add organic matter to the soil and bring them to, help in bringing them to normal position, that's the things. Then, of course, they produce lot of mucilage. They are helpful for soil binding property, that is soil structure. They help improving soil structure. They help in avoiding soil erosion because of the production of mucilaginous substance and so on. They are the source of food for other algal grazers, aquatic life in the aquatic ecosystems, other fauna in both the terrestrial and aquatic ecosystems, represented both freshwater and marine water ecosystems and so on. Like that, you can list so many things. Like go on listing the things, some important features for this. Of course, they are also the source of many important thing, like company, things like, agar agar and so on. So this is some brief information on algae, eukaryotic algae. Eukaryotic algae, of course, the name, they are classified mainly based on the composition of the pigments, primary pigment and secondary pigments. What pigment is dominant, accordingly they are given the names. Okay. So of course, protozoa are there. We will conclude in the next class tomorrow. So Anything you have, you can ask me. Govisidesh.
Sir.
So you have, tomorrow what time we have class? 2:15 to
3:15, sir.
2:15 to 3:15. And one more organism is there. What we will complete tomorrow, that particular thing, and move on to the next chapter.
Sir. Dikshita want to ask something, sir.
Yaro?
Dikshita, sir.
ಹೇಳಿ Dikshita.
Sir, study questions, sir.
Oh, correct. You had asked me. I will, because these days I don't visit lab, but that's the problem because of corona. I will just make some thing, in handwritten things, and I will arrange for the chapter-wise questions. Okay.
Yes, sir.
Okay, and all of you, the request. So the thing, like after that practical which you had on Saturday, 3 weeks back, your some thing was closed, and online classes were initiated because of COVID. And that day you had we had taken the things for enumeration. Okay, till that point, our practicals, you have written the practicals. It is followed by the next practical is isolation, purification, and preservation of the cultures. Next practical, everything we have given, it's for your benefit. Though we don't know whether you will be able to meet and take these practicals before the end of the things. At least you write the practicals. Everything we have given in the form of slides and other visuals and other things with explanation, and we have also forwarded protocols. The next practical, of course, it is on staining techniques in microbiology. In that different things where it's applications and all. And this week I will take up the biochemical activities of microorganisms. Kindly, of course, continue to write the record so that you will have some write-ups in your second year or third year. So the things, unfortunately, of course, if you are fortunate enough, if you come back, we'll complete those practicals. So otherwise, I will send the study questions. Okay, Dikshita?
Sir, what about midterm syllabus, sir? Midterm syllabus, sir.
Oh, midterm. I don't know whether you're going to have online or what they are going to do. Any information?
Sir, almost online, sir.
So I will send you that particular thing, the portion and all that, kind of things, what you're going to have. I told you in one of the classes, I'll split it into, of course, if the
25 and 50% objective.
That's what, that way, true or false or multiple choice or match the following, and the remaining 50% this thing I want to give, but a week before we got struck. So if they permit us to set that kind of question paper, then otherwise if they expect us to give only objective type, of course for your practice I will send it separately. Okay, sample question paper.
Okay, sir.
Okay, otherwise for this thing, like midterm, so the exactly 50% of the portions I will include. So, and I will set you in advance, how you have to prepare. Okay.
Okay, sir. Thank you, sir.
Okay, thank you all.
Thank you, sir. Thank you.
See you tomorrow.